Hi, everyone. My name is Kenny Coogan. I'm the Education Director of the International Carnivorous Plant Society. Thank you for coming tonight to learn about how to grow Byblis with John Ende. I did want to let you know that next month we will be doing our monthly happy hour. Bring a drink, bring a plant, and it's always a good time. We have little breakout rooms where you could talk about lots of different genera and what you're growing and your challenges. Also in March, we will be having a webinar with Drew Martinez from Carnivorous, which is in Austin, Texas. He's going to be talking about grow lights for carnivorous plants. That is March 31st. We are very excited for that talk. On May 4th, we are having our second annual World Carnivorous Plant Day. On that day, on our Facebook page and on our YouTube page, we will be publishing 24 videos, one every hour on the hour. And we have experts from around the world who will be talking about conservation and cultivation. Part of World Carnivorous Plant Day is our annual photo contest. This year is our third annual photo contest. All you have to do to enter is to follow our social media page, and then you can submit up to 5 images. This year, lots of people have only been submitting 1 image, and I've emailed all of you and I've told you that you can submit 4 more images. The judges for this year are the winners from last year, which is exciting because we have representation from France, Poland, and Australia. And the winners, there's going to be 3 winners this year. Each winner receives a 1-year membership to the International Carnivorous Plant Society, which is awesome. Part of World Carnivorous Plant Day, we want you to get excited about these plants. If you go to the website, carnivousplants.org, you'll see that we have a little link on the bottom that says 2022 merch. So you can get our logo for World Carnivorous Plant Day on a t-shirt, on a mug, on a tote bag. You could be the talk of the town. And something else that our members are doing is last August, we had the first Carnivores in the Classroom grant where we were able to provide 24 classrooms with $150 each. And that allowed kids in K through 12 to add carnivorous plants to their classroom. And that is because we had some money from members like you. If you think this is a good idea and you think that kids should be growing plants in their classroom, you can go to carnivousplants.org to donate. And tonight, without further ado, I'm going to bring on John Ende.
Whenever we think of Byblis, I've often heard people describe them as knockoff Drosera, which, I mean, to be honest, is kind of fair. At first glance, they don't really have much different. They don't really seem very inspiring. They certainly are beautiful. Got great flowers. I don't have any in flower right now to show you, but they get overlooked. And because of that, people don't really grow them. So I hope to inspire some of you with this presentation to get involved in the cultivation of Byblis. So in this presentation, I'm going to go over 4 sections, which are going to be the basics. What are Byblis and why are they so rare? Which there's a numerous reasonings for that. How to cultivate Byblis, which is all of my personal research, experience, and all kinds of knowledge that's been shared with me over the years. I'm then going to go into advanced cultivation, how to make a Byblis bush. So something very similar to this guy right here, where you get many meristems growing off of it. Tissue culturing techniques, which are courtesy of Elizabeth from Curious Plant, and asexual cloning techniques for all Byblis species. You can asexually propagate all of them. And then I'm going to go through my Byblis journey. So anyone who wanted to just see the cultivation stuff can dip out and not have to hear on how I learned to grow all these. So here we go. So the basics. What are Byblis? Byblis is a genus of Australian and Indonesian carnivorous plants that utilize sticky traps to capture prey. They're usually found near seasonal pools in a large range across coastal Western and Northern Australia, with populations of Byblis lymphora being in Indonesia. They cohabitate the same environment as Pedularis and tuberous Drosera, as well as Nepenthes and Utriculata. They have a very diverse habitat. The genus can be split into two complexes, Gigantea and Lymphora. Now, one thing I do want to note is that they do have one interesting behavior that Drosera do not, okay? And this is where it's like, ooh. They'll take, and I don't know how well this is going to show up on the camera, they'll take their prey and they'll move it to the underside of the leaf to optimize photosynthesis. They also only digest their prey at night. So the Lymphora complex. The Lymphora complex, the Liniflora complex, contains Byblis aquatica, Byblis cahui, Byblis liniflora, Byblis pilbarana, and Byblis virida. This complex is usually seen grown as annuals in situ, and some populations will grow as perennials if favorable conditions persist in the dry season, which there's been all kinds of really exciting pictures I've been seeing recently, which I might have a future video for ICPS in the works about it, that yeah, a lot of the Lymphora complex might actually be perennial in certain sites in the wild. Moving on to the Gigantea complex. The Gigantea complex contains Byblis lamellata, Byblis gigantea, and these species are observed to grow perennially in situ, demonstrating a summer dormancy period, which is actually really cool and very unique. If you've grown tuberous sundews, you've probably seen this, where they'll die back down in anticipation of a dry season and then start regrowing. And it's something that we usually here in North America never have to deal with, which is a really fascinating process. This is caused by the intensely hot Australian summer, evaporating all the water from the seasonal pools that they grow near. During this dormancy, Byblis gigantea, like I have here, will die back to near its roots, where you can kind of see its growth point down here. And lamellata will lose its foliage, which lamellata grows much more like a bush, but sometimes will regrow from its old stems if conditions improve. Both exhibit growth from the rootstock post-dormancy. So the question has to be asked, why are these plants so rare? They're understudied. They're notoriously difficult to germinate, with one exception being Byblis lymphora. The Lymphora complex suffers from growers not wanting to invest in an annual plant. Byblis germination rates decline significantly after several generations of successive pollination. Self-pollination, excuse me. Byblis have a perception of being difficult to grow due to all of the above. And yeah, it's no wonder why they're not more widespread if you take all these things into account. So they're understudied. They're rarely mentioned outside of any publication detailing, well, Byblis are rarely mentioned in any publication detailing carnivorous plants. Big shout out to the International Carnivorous Plant Society for the video and always having articles on the topic. This ranges from YouTube videos to plant systemic textbooks. And when they're mentioned, they're often referred to as incredibly similar to Drosera, despite having many unique adaptations. Their movement of prey to the underside of the leaf to optimize photosynthetic surface area during digestion being a particularly interesting behavioral difference. It's very awesome to note that they move, and they will move their prey to where they're able to get more sunlight. It's mind-blowing. You don't really see that with Drosera too often. Now, germination. Byblis and their habitats usually germinate after either germinate immediately after dehiscence, or if they dry after a wildfire comes through and degrades the seed coat. And I need to test this ex pseudo because this is kind of exciting new information. It appears that in the wild, they will just germinate right then on the spot as soon as the seed falls, if the conditions are right. And that's kind of exciting because that kind of puts a lot of the fire germination thing on its head. But in cultivation, we generally have to use some kind of treatment in order to grow them, which either gibberic acid or a small fire on top of the soil to simulate conditions if the seed has been stored and dried. The required treatment and subsequent long germination times are discouraging for new Byblis growers. I've had pots, even with germination, take up to 3 years to sprout. So yeah, it's kind of discouraging to grow some of them from seed because you'll plant them if you have some Drosera seed you got with them. In a month, you'll have your Drosera, but it's a crapshoot on the Byblis. It's either going to be, a month to 6, and you never know, which I always recommend. Never throw away a Byblis seed pot. Lymphora complex are seen as annuals. As most carnivorous plants are perennial, Byblis and the Lymphora complex are often looked down as less valuable due to their annual growth when given poor care. This perception, as well as the difficult germination process, has made the genus very unappealing at first glance, which it kind of is unappealing when you think about it. Why would I invest in a pack of seeds that costs, $10 to $15 of a plant that's going to probably die out on me, that requires special germination? And even if I do get it growing right, the literature says it's only going to last 6 months. It doesn't seem as valuable as, say a sundew that you grow and you can have for many years. Oh, that's interesting. So we're moving on into actual cultivation. So we're going to start putting some information out there to kind of turn a lot of that on its head. For their pot, I've grown mature specimens, which you can literally see this guy right here starting to flower. I'll flower this gigantea in this pot right here. And I don't know if you guys can see my camera or not in the recording. Oh, well, either way, Byblis gigantea can be flowered in a 3-inch deep by 2.5 by 2.5 inch wide pot. Pots of pot depth of 6+ inches is recommended for a very large bushing specimen. Byblis can be grown in a wide variety of medium mixes. Sand, peat, perlite can all be mixed together. And I've used just peat perlite, used just sand perlite, just sand peat. Long fiber sphagnum moss works perfectly fine after you've germinated, especially if you're doing cuttings. I highly recommend if you are doing cuttings and then long-term growth, long fiber definitely seems to be one of the better mediums, at least indoors. Got to put that asterisk there. For their lighting, they can handle direct sunlight and can be grown in under T12 shop lights. I found best results produced under a light cycle of 14 hours on and 14 hours off indoors. Lowering to 12 hours on and raising it back to 14 over the course of a month can induce flowering in mature specimens, which is kind of a cool tip I've learned. Red-blue LEDs produce a deep red and purple coloration in most specimens. Blue LEDs produce a very natural green-looking specimen. For watering, Byblis can be tray watered. I recommend top watering larger specimens once a month. Seedlings will likely die if you top water them. A consistent cycle of 1 to 2 inches in the tray, given my pot size at least, and then allowing it to dry over the course of a week or two is key to maintaining the optimal root health. They need that cycle of actual wet, dry floodingness to then dry out. And I think it promotes more oxidation to the roots is what I'm running with my hypothesis on it. For humidity, they appreciate humid environments, so 45% to 80%, but never miss them. That seems to break the surface tension on their dew and allows them and starts allowing a lot more evaporation and leaf desiccation. So I've had specimens where I've missed them in the past, and I've even had people report this to me where they'll miss them or have them under misters, and 2-3 days later it's toasted. And the only thing I can think of is it relates to on how they only digest at night. They're probably very sensitive to evaporation. Transplanting. You should only transplant them as seedlings with as much of the surrounding medium being transferred for best results. You break that taproot on a seedling, it's more than likely dead. Larger specimens can be transplanted if root bound, but no attempt should be made to ever tease the roots. You don't want to break them. The root system takes an immense time to recover if damaged. Plants are likely to die if the root system is damaged. For feeding, Byblis are voracious. They love to be fed often. And whenever I recommend feeding, I use a 1/4 strength Maxi solution on them once a week. And even then, as you'll see in the advanced section, I have moved to having to use soil fertilization because there's that voracious. If you have a fruit fly problem in your kitchen, forget the sundew. They will not do anywhere near the work that a Byblis will do. And I have firsthand seen Byblis, particularly filifolia, just absolutely clean house and take out an entire swarm of fruit flies at one of my friends' houses. It's actually quite impressive. Took it about a week. So for advanced cultivation, how we grow big old giant bushes and all kinds of other cool things. Germination, which is actually kind of sad. This has to be an advanced thing. I recommend a combination of GA3 at 500 parts per million for 12 hours and using either liquid smoke or a smoke bath, or catching the top of the medium on fire for all species when the seed is dried. Liniflora and the exceedingly fresh seed should not be treated, as acid or heat can harm the seed. Germination could take upwards to 2 to 3 years, even with treatment. Never give up on a pot of Byblis seeds, and I mean that. It can take a long time. High humidity can encourage germination, but also greatly excuse me one moment. Sorry, daughter's trying to come see the plants. For pollination, Byblis are bud pollinate in the wild. Using either an electric hand raiser, a tuning fork, or any other vibrating mechanism small enough to reach the anther can release the pollen. Move the pollen to another plant stigma to produce seed. Byblis liniflora can self-pollinate. All other Byblis species have to be pollinated by another specimen. So it's kind of hard to get some really good seed. Some of them, like the other liniflora members, can be self-pollinated, but you will see incredibly quick degradation in your genetics is what I've hypothesized it. Basically, after 2 or 3 generations, you'll see that your germination rates will plummet and you will have very few viable, healthy specimens. That's why I've started going into asexual reproduction. Oh, I got the wrong picture on this slide. Oh, wait, actually, no, that's the right one. All species can be cloned from stem cuttings. I use meristems, so the top growing plate, and count 6 to 7 nodes, places where the leaves grow out down. I remove the bottom 2 to 3 leaves and place them in long fiber sphagnum moss. Rooting will occur within 3 to 7 days for most liniflora complex members. Using IBA rooting hormone on the Gigantea complex members will give you the best results, and rooting will occur within 1 to 2 months. And right there, you can see small little root coming off a cutting. Asexual reproduction, root cuttings. Gigantea complex members in large liniflora bushes produce thick roots. Using 1 to 2 inch segments placed either in moist long fiber sphagnum moss or on moist peat perlite in a very humid environment or inside a bag. And plantlets will form within 2 to 3 months if they don't mold out. So keep a very close eye on that and allow a lot of air circulation from time to time. Now, on tissue culture, and this is awesome information from Elizabeth from Curious Plant. So I personally do not tissue culture. So I asked her to give me some information, and I didn't want to doctor it all, but I'll read it off here for you guys. As far as tissue culture goes for Byblis, we have always used a standard 1/3 MS salt concentration that is typical for a lot of carnivorous plants. The seed is fairly easy to sprout on agar, and the plants grow to a large size quickly. I've noticed some species like Roridia tend to get stuck in callus form more easily than others. Rooted plants have a higher success rate with acclimation than unrooted plants. Byblis cultures tend to degrade after the first 2 to 3 rounds of multiplication, and then successful acclimation rates drop significantly after that. This doesn't necessarily mean that Byblis aren't good candidates for tissue culture, though a large number of plants can still be potted out successfully from those first couple of multiplication rounds. I think one strategy would be continually prepare new cultures with seeds so that older cultures can be phased out before they have time to degrade, which would offer a more steady rate of successful acclimations. Soil fertilization. The best Byblis specimens are usually grown with some level of soil fertilization. Exact ratios are not known yet for best results, but if you go too high, you'll definitely see fungus. I recommend feeding it once a month with diluted solution of Maxi if a larger plant is desired. You definitely can start it very young and then slowly start working up your solution as the plant ages, and you'll get a much more impressive specimen. And as you do soil fertilization, you'll see very weird meristem growth, especially if you do pruning and cuttings. Now, this is an awesome topic that I personally haven't seen, but I love seeing online. Fascication. Byblis can become fascicated if grown in a wide and shallow pot. It seems that the root surface area is exposed to more available water. Well, as the root surface area is exposed to more available water, that fascication is usually observed. This picture is courtesy of ZX Ty. Hope I said that right, from the Byblis Facebook group. And I've always noticed that from seeing pictures of these specimens, they're always in very wide pots, always constantly wet. And my hypothesis on that is that the plant just keeps trying to siphon more and more moisture up to the top, and it leads to deformities. Usually whenever I keep my plants incredibly wet for long periods of time, though, I'll see fungal infections, which is why I've probably never seen fascication. So how to make a bush. An established specimen in a deep pot makes for a great bush. With good fertilization and a strong light, a single meristem will form. When about 2 to 3 inches from the desired height, prune the top. This will encourage new branches to form. When the branches are several inches long, prune them to encourage multiple meristems from the stub branch. And like you can see here, whenever I started taking cuttings off of this plant, every single meristem started bursting, especially after I did some soil fertilization. It was actually very impressive. Which leads us into the last part of this, my cultivation journey. I started growing carnivorous plants in early 2013. After reading The Savage Garden, I was chomping at the bit to try and grow these tiny little Christmas trees. Byblis filifolium, Byblis liniflora entered my collection around September of 2013, which right here, I actually had to do some digging and I found some pictures of my first linifloras. So my first Byblis. My first Byblis were grown in conditions that are terrible for carnivorous plants, which is very shameful to admit, but I admit my mistakes. I used fertilized peat moss from a brand that should not be named and unwashed river sand. Many seedlings died out to dampening off or other fungal infections. And when I transplanted them out of the Pyrex container, and the ones that survived, as you can see from these pictures, they were very vigorous, to say the least. Not a single one of them, whenever you had them stretched out, and a few of them, whenever I just let them go, were over a foot long. And then I had a very particular specimen that I was like, okay, put it outside. So I had a small tray of plants out on my porch, put one Byblis filifolium out there. And over the course of a month, it grew from about 1.5 feet long to this very large, almost 4 foot long stem behemoth that you see in this picture in the bottom right, which produced these very cool flowers. I had this mutated flower over here on the left, you can see. And it is probably the most impressive Byblis specimen I have ever grown. But a heat wave came, plant died out, went brown and dried up. And I was like, okay, toast it, put it off to the side. Several weeks later, rainstorm came through, didn't think anything of it. 2 days later, after I came back out to check on the Sarracenia I had out there, oh, it started to regrow, but then it died because I left it out to dry. And it was actually undergoing summer dormancy, which kind of blew my mind because the liniflora complex is not known to have summer dormancy. So I had that head scratching experience and was just like, okay, whatever. Life went on. Oh, of course, now my pictures are messed up. After my experience with the large Byblis and learning that the carnivorous plants dislike fertilized peat moss, I noticed all my subsequent plants that germinated never reached the same impressive size as their parents. And I wrote it off as, oh, the genetics are degrading for self-pollination. But honestly, I just enjoyed not having the hassle with large sprawling specimens that I would have to keep from crawling over every other plant. And there were many points where for, 3 to 6 month spans where I would just lose Byblis for a while, only to have seeds that, I pollinated out, germinate years later. And the ridiculous wait time for germination really does pay off sometimes, especially when you're learning the school of hard knocks way that I was. So then in 2019, I discovered a jumping spider cleaning off one of my Byblis linifloras, and I was like, okay, that's crazy. Which then blew my mind is that it was moving around and going from plant to plant doing the same behavior. So I wrote Alan Laurie, which great guy, about the remarkable findings, and he noted the spider appeared to have similar smooth exoskeleton to the Stereocopsis beetles that can be found on the Byblis in Australia. Communicating with him about that renewed my passion for the genus, which led me to start digging in a way around the germination process, which was even my personal hang up with why I didn't grow more. So I found several references in the early 1990s of growers having success with Byblis stem cuttings. The Savage Garden even notes that you can have some success with Liniflora and Filifolia. Whenever I was typing in Byblis cuttings in YouTube, it kind of blew my mind though, because there were several videos from Southeast Asian growers propagating Byblis with stem cuttings and long fiber sphagnum moss. No treatment, nothing, just pop them in, and a few days later having roots. And I was just dumbfounded, because why are we germinating these plants whenever you could just have somebody who's skilled, like a commercial grower, germinate them and then just propagate them out so everyone else can keep them going. So over the winter of 2019 to 2020, I started doing stem cuttings of the species I had present, and I had remarkable success. I was able to turn 4 plants into 32 within a few months. It was quite astounding. I noticed one of my species of Byblis filifolia was much more vigorous than the rest of my stock. And this is where my opinion that filifolia, outside of the germination, once it gets going, might legitimately be a better commercial specimen than Drosera capensis for newer growers. I've given out several near maturity specimens, and I've had people beat on them, and they actually killed capensis and kept the filifolia alive, which astounds me. So over the past few years, my cloning success rate grew exponentially. I started to sell and give out Byblis to private collectors to see if they could replicate my success. I started communicating with many nursery owners and private growers about their Byblis experiences. Pictured here is a photograph of earlier this month of a Byblis filifolia I sent to Chris Hardy. And you can see I think he said it was over 3 feet tall. Either way, it's still an impressively large specimen. From those growers, I gathered information on their growing conditions, how tall their plants would get, how long and why they would expire. I gathered all the species except Byblis limulata and proceeded to use stem cuttings to test stem propagation on all of the species, which every time I've tried cloning a new species, it's an immense amount of cringe because I don't know if it's going to work. But I can report that I have had success with every species I have tested. It was kind of a nail biter with Gigantea with a long wait time, but it pulled through. So that leads us to where we are in the present. This past winter, I sadly neglected my collection, particularly the Byblis. The regular watering schedule I had them on made them susceptible to fungal infection. I had a full tray of filifolia die out on me. But throughout all of this, some exciting news. Byblis pilbarana, despite its normally small stature, gave me one specimen that grew way more impressively than the 9 other plants I had. I'm hopeful this particular specimen can become commercially viable. So there's some exciting information there. And with that, that wraps up this section of my presentation. I wanted to leave a lot of time for people to ask me questions and whatnot.
Rochelle wants to know, have you seen any difficulty using GA3?
The only difficulty I've seen is if you don't already have it pre-packaged out or pre-diluted, however, which way you're going, I've seen a lot of people make the solution wrong. And I think that's one of the most common issues, is like trying to figure out how much to dilute and the right ratio of fluid to use to actually make it right. That's where I shy away from GA3 and try to use fire germination when at all possible. But with like the rare species like Gigantea, I always use GA3 because I just want reliability in something so rare.
All right, very good. Somebody else asked about liquid smoke, and I know you did mention it in your presentation.
Liquid smoke is definitely a great recommendation. I personally will just use the fire germination because it's the way I've done it for many years. And, you know, I forgot to bring down the little particular jar used for it, but I basically have a little jar, put a little piece of cotton on top of shot glass inside of it, put the Byblis seed around the shot glass, catch the cotton wad on fire, seal up the mason jar looking thing, and let the smoke trap in there, rinse and repeat that over a few hours. And that's how I smoke or fire treat them. That seems to give me some very good success. However, I think the liquid smoke kind of cuts that all out. So I don't know, I'm just superstitious in my germination method. But I think the liquid smoke is just a perfectly fine alternative.
Very good. When you were showing the picture of your 4-foot tall Byblis, Phillips, no wonder why you got addicted to Byblis. And then he suggests that you write a book.
I'm actually in the talks with all kinds of fun people about writing some fun, interesting stuff. I definitely do want to make a cultivation book one day, but there's some plants I gotta work on first before I get there. Though that bush, oh my goodness, for years that thing intimidated me. Okay, so like people are like, oh, you obviously were hooked after that. No, that thing intimidated me. I had to put it outside because it was leaning on all of my other Drosera, light starving them, and I'm just like, I don't know what I'm going to do with this thing. It's kind of like the Brocchinia I have in here. Like I bought it years ago and it's just been growing so large. I'm just like, I don't know what I'm going to do with this thing.
Or does your setup have fans?
I have several fans. Like right now, you can probably see a bunch of different covers on them. It's because over here in Northeast Ohio, it's incredibly dry throughout the winter. And like right now, yeah, I'm at 40% humidity. I put on humidity domes to try and keep it up. But in the summertime, I have fans going all throughout here. They're all uncovered. If you're over 50% humidity, Byblis in particular, they'll grow great. The big thing though that I think I didn't mention, and I should mention now, is that the temperature, you want to keep them over 70 degrees if at all possible. Once you start going lower than that, or even like the low 70, 72, right around there, you'll start noticing that they'll start slowing down in their growth. And if you keep them there too long, you'll start seeing fungal infections. So keep them around 50% humidity, above 72, and you're pretty good to go.
Very good. Lots of people are saying you did a great presentation. And of course, I agree.
Awesome.
Rochelle says, I tried growing annual Byblis outdoors and indoors using GL, tropical setting, had good growth, encountered mealybugs from those grown outdoors. Have you had any experience with pests and how to prevent it?
So I actually was going to do a slide on pests and whatnot. However, whenever I reviewed the previous presentation, which I'm going to plug that one for anyone who hasn't watched it, please go watch the recording of it. Damon from California Carnivorous did an awesome job with that, talking about all of the different kinds of pesticide. And honestly, he already touched on it that almost everything he suggested, he's also tested on Byblis. And I agree with his suggestions that almost every one of the things he suggested, I've used personally on them over the years. They all work. So you have a lot of options available. If you have mealy, if you have scale, if you have, anything like that, you have just a wide array. And my only suggestion is definitely read your labels. Make sure to apply at night, if at all possible, which even most of them will even tell you on the label you want to do it near sundown, if not after sundown anyways. Even indoors, do it when the lights are off. You don't even want to chance burning them because they have very soft, fleshy tissue, and it's not a fun time.
Blake wants to know, are there any difficulties with transplanting or repotting?
Let's see if I can get lucky with this guy. Oh, I did. Oh, I got really lucky. Okay, I wanted to take a picture of this for the lecture, but basically right here with this specimen, I don't think I'm going to be able to repot him up good again. Trying to focus it in. If you see, I'm not sure if that's not mycelium or not on these guys. I don't even know for sure if these guys are producing all of their own root mass there. And I kind of want to take out my little magnifying glass after this and look at that, because I didn't think I would be able to pull it out without breaking it. Usually whenever you try to uproot a Byblis like this, especially a clone like this, that thing just snaps. However, if you notice, it's incredibly fine hairs all throughout that. You'll try to uproot and try to transplant a Byblis, okay? And whenever you actually get it up, the first few times I did it, I was like, oh, I got the entire root mass, got a nice big thick section of white, and I'm like, okay, it's going great. I definitely think that if it's mycelium, or if it's just very, very fine hair-like projections off of the roots, like that's actually what the plant is using to uptake its moisture, and it's incredibly delicate. It's also the first thing I've noted that if you have root rot in a plant, whenever I've uprooted ones that have just barely started to wilt from it, I've noted that like all of that white filament there, like that really white fuzzy filament or whatever is going on there, is just all decimated first, which definitely indicates to me that's the part that's actually the sensitive bit. I don't know how well it's coming in. Kenny, is it coming in good?
When you hold it closer to your face, it's better.
There we go. Okay, yeah, it's very fuzzy, which it's very unlike Drosera roots or Venus flytrap roots if you have ever uprooted them. And the best results with transplanting, you want to just take the whole pot of medium and try to get it out as solid as possible and try to make as perfect as an indentation as possible. And you'll have the best results with that. That's why once you get them germinated, you'll have like a small 2 to 3 week window where you can get a spoon under it perfectly without breaking the taproot and perfectly place it on another, pot and get it going great. Outside of that window, you're chancing it really hard. Hopefully that helps out with that.
Jacob wants to know what are some good resources to find Byblis to start growing them?
So that's actually quite tough. And it depends a lot on availability. Since this is going to be turned into a video, my best suggestion is just private message me through either my personal email or Facebook, some way like that, and I'll try my best to plug you in with what you're looking for. But they're definitely rare. Private growers have them from time to time, and I try to keep tabs on who has what. There's a few people that offer them commercially, and that's hit or miss and very rare. Just like I was saying, like it's kind of hard if people are doing it from seed to keep them going for more than like 3 or 4 years. So I'm hoping more people start asexually reproducing them like I am, because that'll definitely make them more available. But yeah, I know of a few growers right now in the US that have them. There's a few people internationally that I know who are offering them, but it's very sad because Alan Laurie was the man for Byblis, and he definitely made sure that every site he could was available. And ever since his passing, it's been kind of a void. So I imagine it's going to be, a few years before that kind of gets plugged. But in the meantime, I'll try my best to help you out.
Do you currently have all of the species that are represented?
I am missing Lamellata, and another exciting bit that is kind of, hush hush, even from Alan, like Alan even knew himself that there is quite a few other species that could be described that the taxonomical work hasn't been done on. I currently, in my cultivation of the known species, I've never been able to find Lamellata. And I know of a few growers who have it, and I've been trying to sweet talk them into giving me either seed or a cutting or something. I'm just like, take a cutting, I'll grow it. But yeah, no, even for me, some of the species are incredibly rare. And yeah, Pilbarana, good luck on that one. That one's a really tough one to find too.
Don Schiller says, great information. I am new to Byblis. He has gigantea and it's growing well. The liniflora isn't germinating at all. Would smoke work after GA3?
Yes. And another thing that I can suggest in that, if it hasn't germinated and it's been a couple months, okay, I've had this particular situation happen with me with a few of mine that I've been like, oh man, I want that thing to grow. You can still do a quick round of fire on top of it, like take some paper, crumple it up, make sure the soil is still wet. Do a round of fire. You can soak it with liquid smoke. I've done that before and had success. But ultimately, after seeing like the newer information I've been seeing from actual habitat pictures and reports, I think it comes down to you just need to be able to soak that seed enough for the moisture to get in through the seed coating, especially like if you have the smoke, liquid smoke, like put it in, make sure you have it in a flood, like you have it in a pot, flood it out so none of it's getting down, flooding through the soil. Make sure it's sitting up on top, really soaking into those seeds. Because it seems like once you're able to penetrate the seed coat, you get way better germination results. And I think it's actually a misnomer. I think what's actually happened with liniflora is people have just gotten really fresh seed. Because I've noted that, like, I've had liniflora seeds before take many months to germinate just out of the blue, and some of them just always germinate immediately. So who knows? Maybe it's just liniflora has been more observed to do the thing it does in pseudo.
So Philip has a question, and we're kind of wrapping up. So if anyone else has a question, feel free to type it. So Philip has a question, and I've been talking to you, John, for like the past month. So I think I almost know the answer. He said it would be nice to have a network of registered clones with third known lineage so folks can keep the genetics diverse. But I think, John, you told me that you can't find the registered plants.
If anybody who is listening to me can find or knows of anyone who is growing right now, any of the old registered cultivars, please let me know. Like, I know people who have seeds, but that technically doesn't make a true cultivar anymore. It means you have the genetics still somewhat available in that, but it's not a true cultivar. So to me, it's like until somebody comes out of the woodwork and proves me wrong, I have yet to see one of the previously registered cultivars. As for the genetic lineages, I do have location data on all of my plants. So I do know the location of mine. I know a lot of other growers do too. And I hope that tradition stays alive. And especially like if we're starting to clone it, I think it'll be a lot more readily available. Like people will be like, oh yeah, now I have this one that I can clone off and trade with somebody else. It'll make the sites a lot more, like a trading card as opposed to, oh, now I'm just exclusive because I'm the only person who grows aquatica.
Very good. All right. We have one last question or one last comment from Rochelle. She says, I agree with how difficult it is to keep up with the cross-pollination to produce seeds continuously. Proper timing is needed to pollinate. Looking forward to trying out gigantea. She really likes the white flowered variety.
I honestly have only seen one white flower Byblis in my own personal cultivation, and it was a gahui that died almost immediately. So I'm right there with you. White flowered Byblis are awesome. And yeah, that's why I started doing the stem cuttings, which I'm not kidding. It's one of the easiest processes ever. If you are growing a Byblis right now, get some long fiber sphagnum moss, just like that, just snip right off, toss it right in the moss, done. It will root within 3 to 5 days. It blows my mind. It's almost as easy as pings, which a funny thing was like, so I realized something. What people need to get in the habit of doing with Byblis, we need to start doing it like on how we think of a ping. So pings are known to rot out a lot and very frequently, and most pings will expire within a year to 2 years if things aren't perfect. We'll just be honest here. I think a similar situation with Byblis, like we need to have the mindset of, oh, I need to take a stem cutting every 3 to 4 months, and you will once you start doing that, I have not lost any of the genetics outside of Aquatica. I'll admit that. But outside of that, every other genetic lineage I have had, I've kept, especially of the quote unquote annuals now for over 2 years. And it's really awesome to have that, because now it's like, okay, I don't have to go looking for seeds again of that. I just have it.
Very good. We want to thank you, John, for doing a wonderful presentation. We all agree that your enthusiasm is inspiring, and we hope that more people start growing them. Lots of comments are coming in now saying that the talk was super interesting and very helpful.
Awesome. I look forward to it. Sorry for the background noise and interruptions throughout, but I hope you guys found it all enjoyable, and who knows, maybe it inspires you guys to grow some Byblis.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.